So we're going to continue on with these molecular aspects of genetics. We're going to move on from replication of DNA to actual transcription of genes. This is the process of turning genes into actual protein products and actual carry out some of the functions that these genes have within the cell. Here's the chapter outline for your perusal and for your reference for when you are studying. There are multiple different types of polymerases. We talked about DNA polymerase 1 and DNA polymerase 3 in the last chapter, which is responsible for some of the replication aspects of taking one chromosome and making it into another chromosome for the process of mitosis. But there's also polymerases that are responsible for transcribing genes and turning them into those protein products. And so these polymerases called RNA polymerase. This is an example of a crystal structure of RNA polymerase here on this slide. And so this is one of the molecular aspects that we'll be talking about in this chapter of how we actually turn genes from a blueprint, so to speak, on the chromosome into actually performing function within the cell. So back when these genome projects started kind of unfolding in the early 90s and progressing throughout the 90s, we started to sequence the DNA of some of these smaller organisms. For instance, E. coli had its genome sequenced, and we found that there was 3,200 genes in E. coli. And so when scientists and geneticists at the time looked at humans, the kind of prevailing theory was that, okay, humans are way more complex than this little tiny bacteria E. coli. And so it's got to have much more genes, right? So more complexity must mean that there are more genes underlying it. And so we knew that a simple plant like Arabidopsis, a little tiny plant that I actually do research with in my lab, it had roughly low 22,000 genes, somewhere in that ballpark. And so scientists were asked to place bets on how many genes were in the human genome because we knew some of these lower organisms. And so some people bet very low, roughly 25,000 genes, and then others bet up to like 150,000 genes, right? Because humans are much more complex and we're much more special than stupid Arabidopsis and E. coli. Well, it turns out that humans do have roughly the same amount of genes as E. coli. 26,000 genes roughly. It depends on annotations change over the years. But really, there's very few or about the same amount of genes as some of these organisms we view as much less complex. But what was crazy about it is if we looked at the number of proteins that were also in humans, 25,000 genes roughly, 100,000 proteins is what we would be able to characterize in humans. So how is it possible that something as few as 25,000 genes could lead to as many as 100,000 proteins? And this was a very difficult question for scientists to answer for a long time. And so we knew that DNA was encoded, or proteins were encoded by DNA, by genes. But we didn't know at the time was that a single gene can actually encode multiple proteins. And so genes are made up of introns and exons. So exons are portions of a gene that are actually turned into mRNA and then eventually into protein. Introns are these regions kind of in between genes. We used to call them junk DNA. They actually found that they have regulatory processes and some other functional aspects, so they're not junk really, but they play a role. But there are these kind of spacers in between exons, and so when genes are transcribed, we can have this process called alternative splicing. And what alternative splicing is mix matching the exons from a gene to make different protein products. So say you have 5 exons, 1, 2, 3, 4, 5. One protein may take exons 1, 2, and 3. Another protein product could take 1, 3, and 5. A third protein product could take 2, 3, and 4, etc. So mixing up the exons in genes to make different RNA, mRNA, that will make different protein is how we get from something as small as 25,000 genes to 100,000 proteins. So transcription is the process of taking a gene, and we have this transcription machinery, this RNA polymerase, that takes that information from a gene and it turns it into an mRNA molecule. And so this is kind of the message, M being messenger for messenger RNA, that is sent to the proteome to be made into a protein product. And so the first step is to actually copy and transcribe this information into a strand of mRNA from that DNA blueprint. And then in prokaryotes, the mRNA is immediately converted into a protein. So translation, the process of taking a mRNA molecule and turning it into a protein, occurs simultaneously with transcription. In eukaryotes, we have some processing that occurs, this alternative splicing that I talked about in the last slide, that occurs before it goes to the proteome, so we can turn those 25,000 genes into over 100,000 proteins by doing that mix match process that I talked about. So throughout this chapter, we'll get a little more in detail about how this process occurs. So in early experiments with mRNA, scientists were looking to uncover what the role of mRNA is, where it was located, where it was going, etc. And so in this experiment called a pulse chase experiment, cells were briefly grown in a radioactive uracil media. Remember, with mRNA, uracil takes the place of thymine. And so instead of ATGC, which we see in DNA, in mRNA, we have AUGC, right? So uracil taking the place of thymine. So in these cells that were grown in radioactive uracil, we have mRNA that is going to have A, U, G, and C with a radioactive U. And this is a way that we can label the uracils, but not label the thymine from DNA. And so once those cells were grown there very briefly to label those, or to integrate those uracils into the mRNA, those cells were then taken out of that radioactive uracil media and placed in just normal media. And what we ended up, or what the scientists ended up seeing, was that right after removal from that uracil, radioactive uracil media, we could use, again, those like a kind of Geiger counter or a radio-labeled paper where it would show us where radioactive material was, and we'd see that all the mRNA was in the nucleus. And then after a certain amount of time, those mRNA that were radio-labeled previously from that radio-labeled media, they start to migrate out into the cytoplasm. So they're no longer in the nucleus. So they have this mobile component to them where they originated in the nucleus, but then they spread out into the cytoplasm of the cell. So one thing I want to point out with the difference between RNA and DNA. So RNA has a ribose backbone. DNA has a deoxyribose backbone. So ribose, as you can see here, has an extra hydroxyl group where there is normally just a hydrogen on deoxyribose. And what this allows is for RNA to form more complex structures than DNA. DNA, you find it normally in a double helix. That's all you find it in the cell. RNA can form many hairpin loops and all these weird structures because of this ribose backbone as opposed to DNA. And that facilitates a lot of the functions of RNA, with it being able to have roles inside of enzymes and as messenger RNA, and we're finding regulatory roles of them with genes now. So it gives it a lot more flexibility than DNA, who's the only purpose of DNA really is to preserve those genes and that message to pass on to the next generation. RNA has a much more substantial role in that it does a wider variety of things than what DNA does. So just like DNA, RNA is composed of 4 ribonucleotides. And similar to DNA, we have adenine, guanine, and cytosine. However, unlike DNA, as we talked about a couple slides ago, instead of thymine, we have uracil as the 4th nucleotide for RNA. There are really two classes of RNA molecules. We have messenger RNA, which are the RNA that are transcribed from genes within those chromosomes and sent to the proteome, where they are used to make those growing polypeptide chains, which are eventually turned into proteins, which eventually have some sort of role within the cell. The other class of proteins, or of RNA, are functional RNA. These don't encode information to make proteins, but they have separate roles within the cell. One example is transfer RNA or tRNA. These are molecules of RNA that are responsible for bringing the correct amino acids to the proteome to incorporate into this growing polypeptide chain to make some sort of protein. A second type is ribosomal RNA, rRNA, which are the major components of ribosomes, which are the large macromolecules that are there to guide the assembly of proteins. We have small nuclear RNAs, or snRNAs, which process RNA transcripts in eukaryotes. We have microRNAs, or miRNAs, which regulate protein products and the amount of protein product creation. We have siRNAs, small interfering RNAs, and this is kind of a viral protectant, but it can be harnessed for molecular techniques, which we'll see later in the quarter. And then lastly, we have Pee-wee interacting RNAs, which inhibit transposable elements, which in large abundance could be very dangerous, but also bring necessary genetic diversity to organisms. So we know that DNA has two strands, and generally only one strand of that DNA serves as the template for RNA. So in this example on gene number 2 and 3 here, the gene is read on the 3 to 5 strand in the 5 to 3 gene direction. However, on the opposing strand in gene number 1 will have been read in the opposite direction. So as you can imagine, because all genes aren't palindromes of one another, the direction in the strand is important for transcription of the gene. So only one of the strands is read for transcription to the gene product, but depending on the gene, it can be either of the two strands. Here we have an overview of transcription of two genes in opposite directions. Gene 1, over on the left here, is transcribed in the right to left direction here. So RNA polymerase is proceeding from right to left, and it's reading the lower strand and proceeding in the 5' to 3' direction, while gene 2 is reading the upper strand over here, and it is proceeding also in the 5 to 3' direction. So just like DNA, the template is read in the 3 to 5 direction, but the new molecule is created in the 5 to 3 direction. So here is a double-stranded sequence of DNA that we have anti-parallel to each other, and we have a transcribed piece of RNA, which is on the bottom here. And as you can see, the transcribed piece of RNA is from the 3 to 5' direction of this strand, and it is the complement of every base pair. So the opposite of a guanine is a cytosine. The opposite of a adenine is, remember, this is uracil instead of thymine, because mRNA uses uracil in place of thymine to increase the secondary structures that it can make for the purposes of function that we'll get into later. And then we have a G paired with a C paired with a G, etc., all throughout this entire gene. Briefly, we're going to hit on the stages of transcription here. This is kind of similar to DNA replication. There are slight differences. You'll see a lot of similarities between transcription and DNA replication, but there's also a lot of differences. So first we have initiation. That's where the RNA polymerase binds to the promoter upstream of the gene to create that mRNA transcript. We have elongation. That's where the transcription bubble is formed. So those two DNA pieces, the two strands, have to pull apart to allow that transcription machinery in between to transcribe that gene. So this bubble is formed and it opens that double helix, and then RNA polymerase moves down the DNA to simultaneously create that mRNA molecule. And then lastly, we have termination here. And this is where transcription proceeds past the end of the protein coding sequence. And it creates what's called this UTR, which is this untranslated region. In the case of at the end of a gene, that's the 3' UTR. Remember, it reads from 5 to 3. And these nucleotides at the end in the untranslated region tell the polymerase to stop making mRNA and to kind of fall off or that the gene is over with, so to speak. This is a generalized structure of a gene. So we have the promoter region. So this here is the region where the transcription machinery will recognize. It will kind of lay down, and it's kind of like the starting line of this process of turning a gene into mRNA. And so there are these common elements within a promoter. We have the -35 region and the -10 region. Mind you, this is in E. coli, this is in prokaryotes, not in eukaryotes. But we have this -35 and this -10 region, and then there's usually 15 to 17 base pairs in between them. And these are consensus sites that hold very similar sequence identity. So if we look at the boxes highlighted in yellow here, we'll see that there are these kind of conserved nucleotides generally in these different genes here within E. coli. And so these are recognition sequences where the polymerase, the RNA polymerase, knows that that's where it should attach. So these are kind of this flag being wove and signaling the transcription machinery to come in. That's where it needs to lay down to start transcription of this gene. Now, beyond that, we have this region here. This is the 5' UTR. So this region is turned into an mRNA molecule, but is not transcribed into a protein product. It's kind of this buffer between the promoter and between the actual gene. And then we have this ATG start site for transcription. And then all of these nucleotides here will be turned into, read as codons and turned into the growing polypeptide chain. And then lastly, beyond that, we have this 3' UTR where there's some sort of recognition sequence for that transcription machinery that the gene is over with and it needs to stop. So RNA polymerase is a transcriptional enzyme that is made up of several subunits to make up the whole. So we have alpha subunits, and the role of the alpha subunits is to promote the interaction of regulatory enzymes or proteins with the transcription machinery. So that's kind of a regulator of how much of this to make and how often should we make this transcript. The beta subunits here actually physically interact with the DNA, and they are catalyzing the additional bases being added to the growing mRNA transcript as transcription proceeds. We have this omega subunit, and so this is part of it that a component that regulates. It's kind of like an on-off switch for turning transcription on and off. And then lastly, we have this sigma subunit, sigma 70 it is indicates here. And as you can see, this is the component that is actually binding to that promoter region, the -35 and the -10 unit. And so these three different subunits here, alpha, beta, and omega, those three subunits are the same for all RNA polymerase. And the one thing that differentiates RNA polymerase from binding from one gene to the other is the activity of this sigma subunit. So each sigma subunit is specific to whatever gene is going to be transcribed. So that's the part that says transcribe a heat shock protein instead of a ubiquitin or some other enzyme for that matter. Now, as soon as all of these things are in place and transcription's been initiated, then that sigma subunit, that takes off because that's only there to direct that transcription machinery to the gene that needs to be transcribed at the time. So once it's in place and starting down the road of transcription, it takes off, and then alpha, beta, and omega are there to continue the transcription process. So here we see the transcription machinery. So all those subunits are kind of wound into this egg. A lot of times throughout the textbook, they'll denote the transcription machinery as this little egg going through. We know it's made up of different subunits, but for just simplicity, it's kind of denoted as a single egg here. So as this is proceeding, transcription, the polymerase will unwind the DNA as it goes, and then behind the preceding transcription machinery, the DNA is rewound. So unlike DNA replication, where those strands are pulled apart and then they stay pulled apart and then their complements are built and you end up with two strands, in transcription, the DNA is just pulled apart temporarily just to give that transcription machinery access to that gene temporarily to make that transcript, and then it closes back up. And then if it needs another transcript, it'll repeat this process over and over again. So this will proceed until it gets to this UTR region where this sequence will form this hairpin loop. So like I talked about earlier, because of the presence of uracil, RNA makes these weird structures when they're because they're single-stranded. Unlike DNA, it only really forms a double helix or double-stranded pieces, but RNA forms all these weird substructures. And so in the 5' UTR regions, there are the sequences that will lead to things like hairpin loops. And when that hairpin loop has been created, that is a message to the transcription machinery that it needs to release off of the DNA because there's no longer gene beyond that. That's the end of the gene. Here's an example of the consensus sequence at the 3' UTR of a mRNA sequence. And so as you can see, there is kind of this section of nucleotides who has another section of nucleotides that is completely complementary to it a little ways away. So we have 1, 2, 3, 4, 5, 6, 7 nucleotides on this side. Then we have a sequence of 5 nucleotides. Then we have the exact match to those 7 nucleotides a little bit later. So this forms that secondary structure called a hairpin loop, and that is what leads to the termination of the RNA polymerase that is proceeding down that DNA to transcribe it, and that kind of knocks it off or proceeds to stop transcription of DNA from that point. Let's compare prokaryotic and eukaryotic transcription and translation. And so remember some of the difference we talked about with prokaryotes and eukaryotes. Prokaryotes, the DNA is circular. It's not in a nucleus. It's not compartmentalized like it is in eukaryotes. And we'll also see that there's some difference in the transcription and translation process. So we'll start here with prokaryotes, who, like I said, have a circular DNA chromosome. Transcription of that chromosome occurs all within the cytoplasm because it's not subcompartmentalized into the nucleus like it is in eukaryotes. And so we have transcription of this mRNA molecule happening in the cytoplasm here. And what's interesting with prokaryotes is that because there is no compartmentalization, so there's no need for the mRNA to leave a nucleus because there is no nucleus, the actual transcription and translation occur simultaneously. So the translation machinery, the proteome as we've called it earlier, that will come in and start to read and add to this growing polypeptide chain, which is indicated by 2 little circles here. It'll start to extend that polypeptide chain, reading the mRNA as it's still being transcribed from the DNA. And as it continues on, you'll see that it moves up the mRNA and will continue to add to that polypeptide chain and add to the polypeptide chain until it gets to a finished product. Now eukaryotes were a little bit different because we have compartmentalized DNA. So it's in the nucleus. It's not floating around in the same spot where the proteome is. And so here we have our chromosome that is opened up and ready to be read and transcribed. Now transcription will occur. There's no picture of, I'll draw, here's our That's our transcription machinery there. And it's reading the DNA and starting to make this mRNA. And now what's different from eukaryotes to prokaryotes is that there's that processing aspect. So earlier I talked about how there are these little regions within a eukaryotic gene called introns. And those used to be referred to as junk DNA or non-coding DNA. They have regulatory aspects that we know now, but they're not turned into a protein. And then the portions that are read and turned into a protein, those are called exons. Well, in this processing step, all the introns are spliced out via the spliceosome, and the exons are pasted together to form this mature mRNA molecule. Those mRNA then are transported outside of the nucleus to the cytoplasm, where translation can proceed to read that mRNA and grow the polypeptide chain to make a mature protein product. So we had talked previously about eukaryotes and how in eukaryotes there's this recognition site, the -10 and the -35 region, and there's those consensus sequences there that the transcription machinery binds to. So now let's look at this in eukaryotes. So in eukaryotes there is a region called the TATA binding region. So it's this recognition sequence of TATA. And we have this protein called the TATA binding protein, or TBP for short. And so what happens is this TATA box that I kind of indicated up here is this recognition site in eukaryotes. And so TBP will find and bind to this TATA box, and then this TF2D, transcription factor 2D, will then be recruited with that TATA binding protein to the initiation site for the gene. So once there, that TBP, that TATA binding protein, will recruit other transcription factors to that start site. So we have TF2F, TF2H, TF3, but it will also recruit RNA polymerase, which remember was that holoenzyme that had multiple components to it that we said we would denote as an egg from now on. So this is a very elongated egg, but that was that transcription machinery made of alpha, beta, sigma, and omega groups, and we'll recruit that to the TATA spot. And eventually, once this carboxyl tail domain, so one thing we talked about how this transcription machinery is denoted as an egg. Well, it's really an egg with this real long tail coming off called the carboxyl tail domain. And eventually, throughout this process of initiation, this carboxyl tail domain will start to get phosphorylated. So these P's are just phosphorylation that is occurring on that carboxyl tail domain. But once that occurs, that releases this RNA polymerase from this initiation complex that we had seen formed here at the TATA box, and that'll kind of send the transcription machinery down the railroad track of the gene, so to speak, to start transcribing that gene into mRNA. So in prokaryotes, transcription and translation occur at the same time. So as that mRNA is being extended, the proteome comes in, reads it, and starts making it into a protein. Well, that doesn't happen in eukaryotes because, remember, the transcription is happening in the nucleus, translation happens in the cytoplasm. There is this co-transcriptional processing of the RNA that's occurring. So that is that the mRNA, as it is being transcribed, is also being spliced alternatively with different introns and exons being removed and left in. So in this process of co-transcription of RNA, we have these capping enzymes that come in during the transcription process, and the role of these capping enzymes are to alter that first nucleotide in the growing mRNA to kind of make it a beacon or something that stands out, and we'll get to why that is later. Once that first nucleotide is turned into a cap for this growing mRNA, this RNA splicing machinery will come in and take its place. Now, the role of this RNA splicing machinery is to read those introns and exons, and it will take out all the introns. Remember, those are not transcribed or translated into proteins. And actually, they will also read the exons of this mRNA chain, and that's where this alternative splicing comes in. So these RNA splicing machinery, the spliceosome, will determine which introns are incorporated and which exons are incorporated. So different exons can lead to different protein products, and that's all done by the spliceosome. Lastly, these enzymes come in whose role it is to kind of terminate the growing mRNA chain. And what they do is they add a poly A tail to the end of this mRNA chain. And so a poly A tail is just what it sounds like. It is poly, meaning multiple of something. A is for the adenine, the nucleotide. So it adds a whole bunch of adenines, A, to the end of this mRNA chain. And so now we have something that signifies the beginning, the 5' end, which was that capped altered nucleotide. And then we have something that indicates the 3' end, which was that poly A tail. So our finished product here is a piece of mRNA that has a capped 5' end and then a poly A tail on this 3' end. Now the role of the cap is kind of, like I said, it's a recognition site, and this helps the mRNA be transported out of the nucleus and into the cytoplasm, where it will then be translated into a protein. And the role of the poly A tail is to provide a bit of protection from degradation and breaking down of these mRNAs. Now, one thing to talk about that I don't believe I've mentioned is there's a difference in mRNA and DNA, or RNA in general and DNA, in their longevity. Now, DNA is a much more stable molecule than RNA is, and that's because the role of DNA is kind of to preserve the historical blueprints of how this organism should be. So its role is to pass from generation to generation what genes are involved in this organism, right? It's kind of like the master plan of the organism. Now, the role of RNA is just to be a message for just a quick second, right? And so for RNA transcription, mRNA in the process of transcription, it's only there to, at the moment of transcription, send that message to the proteome to make a protein, and then its job's done, then it's broken down. And so the structure itself is just not as stable as DNA, because it's only meant to be around for, you know, a short while before it's broken down. So this poly A tail is to help it stay around just as long as it needs to without being corrupted from reactive oxygen species and things within the cell to get to that proteome. And then once it finishes its job there, then it is broken down and becomes nucleotides again to become the next piece of mRNA. So when we talked about alternative splicing, I had mentioned that different exons are incorporated into a mRNA to make different proteins. And so here's an example of in eukaryotes, and remember this only happens in eukaryotes, because in prokaryotes there's no spliceosome, there's no compartmentalization into the nucleus, so this is only a eukaryotic trait. So in eukaryotes, here's an example of muscle, or this specific gene, and what it can do is, depending on which exons are alternatively spliced into the mRNA product, we have different protein products. And so first, if we incorporate exon 1A, so I'll highlight here, exon 2B, exons 3, 4, 5, 6B, 7, 8, 9A, and 9B, we get the products of a protein incorporated into striated muscle. Now, the same gene, if instead we incorporate exons 1A, 2A, 3, 4, and 5, 6B, 7, 8, and then 9D, we get the protein that is incorporated into smooth muscle. And this can be very vast in the roles of these. So the first two examples are muscle, but if we look down at this fifth example down here, if we incorporate exons 1b, 3, 4, 5, 6b, 7, 8, 9c, and 9d, we have a protein that is a constituent of brain tissue. So that is the onus of how an organism with 26,000 genes or whatever the current count is for humans. I'm not a human researcher, so I'm not completely up to date on the number. But 26,000 genes can lead to well over 100,000 different proteins because of this alternative splicing phenomenon. So when we talk about alternative splicing in the spliceosome, there has to be some sort of way that the spliceosome knows the boundaries of introns and exons. And that is through these conserved sequences that are in the gene and are transcribed into the mRNA. And specifically, a lot of these splice sites are indicated by this AGGU pattern. So if you look at both of these two splice sites here, they don't indicate the U here, but you can imagine there's a U here. This AGGU, the spliceosome reads that 4-peat of DNA and knows that it has to cut between the GUs and that the introns then are after an AG and then if it's preceded by a GU. So now let's look at how this actual splicing occurs. And so remember the boundaries of these exons and introns are indicated by these consensus sequence, AG, GU. And then we have two small nuclear ribonucleoproteins. So on this this figure here, the book is incorrect. This is not a SNP. So SNPs being small nucleotide polymorphisms. We talked about this. So these are SNRNPs. So these small nuclear ribonucleoproteins initially attach at these two conserved sequences. So U1 will attach at the AGGU, the first AGGU upstream within the intron. The second SNRNP will attach to this consensus adenine that is 15 to 45 nucleotides upstream of this last AGGU region. Once those two SNRNPs are bound, 3 additional SNRNPs will be recruited, U4, U5, and U6. And what this complex does is it will bind to U1 and U2 and start to bring them together. And it kind of forms the precursor of a one of those hairpin loops, kind of like we saw in RNA. So once U5 and U2 are brought together, so this yellow one here is U5. Once they're brought together, U1, who is originally part of that AGGU recognition site, as well as U4 and U6, who were brought in to kind of bring this loop together, they peace out. They're out of here, and all we're left with attached still is that original U2, who was bound to that consensus adenine, 15 to 45 nucleotides before that last AGGU site, and then U5, who is kind of taken over as the indicator of the beginning of the intron. And so what happens then is U5 will help to detach the GU from that first AG, and it will bring it back to form this hairpin loop with the final boundary between the intron and the next exon. So it forms what we call, for some reason, a cowboy's lariat. So I'm assuming that is like a lasso. I'm not much of a rancher or cowboy by any mean, but it forms this the shape what they call a cowboy's lariat, and allows this free hydroxyl group to be exposed on that preceding exon. Then a second splicing reaction occurs from that U5 and U2 snRNP complex that had formed previously, and it allows this second exon to be freed up, and the previous hydroxyl group here is able to form a bond with that second exon, and this intron then is just released out of the mRNA, and we have two exons now that are joined together. So previously in this chapter we talked about functional RNAs, who were RNAs that, unlike mRNA, aren't actually turned into gene products, but are instead used for some sort of molecular role within the cell. And so here we're going to talk a little bit about one of these, which is miRNA, and how they are playing a role in regulating transcription of different genes. So in this example, we're going to talk about a protein called Lin4. Lin4 produces a long RNA molecule that is complementary to itself in the middle. So again, we see this familiar hairpin loop structure here at the top, and it causes the creation of this double-stranded piece of mRNA. Now, once that mRNA is processed from the spliceosome, as we talked about in the last couple of slides, we have pretty much a double-stranded piece of mRNA with a hairpin loop. That double-stranded piece of mRNA is then exported outside of the nucleus into the cytoplasm, where this protein complex called dicer recognizes double-stranded pieces of mRNA, and it binds to that double-stranded piece of mRNA. From there, dicer processes that mRNA, removes the hairpin loop, and leaves about a 22 base pair segment of double-stranded mRNA, which is incorporated into a protein called RISC, or the RISC complex. The RISC complex will then separate that double-stranded RNA into a single-stranded RNA. So now what we end up with is a protein that has a piece of single-stranded RNA that was 22 base pairs in length, roughly, and matches the sequence of DNA that we saw in this original Lin4 gene. So now we have this RISC complex who has a specific sequence, 22 base pair recognition sequence. And what it's doing is it's floating around, and it's within the cytoplasm looking for a piece of mRNA that has that complementary sequence of 22 base pairs that it can bind with. So while it's searching around, if there is a piece of mRNA that is in the process of being translated into protein, it will bind to that piece of mRNA, and it will repress translation of that mRNA into a protein. In addition, that complex will also set in motion the degradation of that mRNA, starting at the poly A tail, so that mRNA will not continue to take up spots in the proteome or try to be transcribed later on at another time into a protein complex. So this Lin4 miRNA gene, it actually has consensus sequences for two other Lin genes. So in this case, Lin14 and Lin28 have this same 22 base pair sequence within the gene. And so transcription of this Lin4 microRNA gene is a way to stop the transcription and translation of these other two genes. So this microRNA, through the dicer-drosha RISC complex, we didn't really get into drosha, but it's another component of this dicer complex, it can turn off other genes. So this microRNA has a role in regulating transcription of the other genes within the cell. So we just talked about miRNAs, which are microRNAs. Now we're going to quickly talk about siRNAs, which are small interfering RNAs. And so the function of these siRNAs is to provide protection from viral infection. And so when a virus dumps its genes into a cell, the genes can be inserted into a host's chromosome, and the direction of those genes could be either sense or antisense. And so you could get transcripts that are occurring complementary to one another. Now, when you have these two complementary strands of mRNA, that will lead to the formation of double-stranded RNA. And so, again, just like in the previous example, that recruits dicer, which is an enzyme that is specifically there to break down this double-stranded RNA. Dicer will recognize that double-stranded RNA and chop it up into these small interfering RNA segments, who are then, just like in the miRNA case, incorporated into a RISC complex, which is a protein recognition complex, who will use half of that double-stranded RNA to look for complements, and it will search out for these target mRNAs who have that complement sequence. So this is a way that it can search for those viral genes that have invaded the cell and make sure to degrade them and not allow them to be used to reproduce viral heads and things of that nature. Once those viral mRNAs are found, they're broken down into or chopped up into little segments where they won't be transcribed and they can't form a full protein product, thus protecting the cell.